who are not AGT members, please consider joining the organization so that AGT can continue to be the voice of the technologist in the laboratory community and be able to offer programs like this in the future. Please check AGT's website, www.agt-info.org, to see the many benefits of membership. Speaking from personal experience, I will share that AGT has provided numerous educational opportunities that have been critical for growth in the technical aspects of laboratory work, particularly in the initial stages of my career. In addition, as I've become more engaged in the organization, I have benefited greatly from the networking opportunities that have emerged. Please feel free to reach out to me offline if you have any questions about how the organization can be of service to you. A quick housekeeping note before we get to today's presentation. Please feel free to ask questions during the session. In the upper right side of your computer screen, you will see a control panel. In the lower portion of that panel, you can type a question or comment and submit it to the webinar organizers. You can do this at any time during the presentation. We will reserve about 10 minutes at the end of the presentation for questions and answers. If you are participating as a group, please designate one person in the group to complete the online evaluation. In order for everyone to receive their certificates of attendance, that person will need to enter the names and email addresses of each person who is participating at your site today into the evaluation. As a backup measure, a sign-in form can be completed and emailed or faxed to AGT as well. Certificates will be emailed to participants approximately 30 days following the webinar. Today, Pamela Althoff and Mary Lou Nelson will address utility and logistics of fish testing for solid tissue tumors. Please allow me to introduce you to our two speakers, both of whom come to us today from the Human Genetics Laboratory at the Monroe-Meyer Institute for Genetics and Rehabilitation at the University of Nebraska Medical Center. Pam Althoff is a laboratory supervisor specializing in fluorescence in situ hybridization for the laboratory. She completed her master's degree in genetics at UNMC in 2008 and maintains an ASCP Board of Certification as a technologist in cytogenetics. Pam began her 15 years of service specializing in probe development, fish, and conventional cytogenetics of solid tumors before expanding into characterization of hematologic malignancies. Pam has 30 publications or presentations at national or international genetics or pathology conferences. Mary Lou Nelson is a laboratory supervisor specializing in solid tumor cytogenetics and molecular genetics for the Human Genetics Laboratory. Mary completed her master's degree in genetics at UNMC in 2008 and maintains an ASCP Board of Certification as a technologist in both cytogenetics and in molecular biology. During her 27 years of service at UNMC, Mary authored or co-authored over 50 peer-reviewed publications covering a range of topics, including conventional cytogenetics and fish of solid tumors and hematologic malignancies, molecular genetics, and research in primate genetics. I'm now going to turn the presentation over to Mary Nelson, who will get us started today.
Thank you, Jennifer, and thank you to the organizers for inviting me to talk to you today. During the course of this webinar, we will address some of the nuances of fish studies on solid tumors, specifically as they relate to paraffin-embedded tissues, touch preparations, and destained metaphase spreads. Hopefully, you will find the information useful on how we optimize hybridization quality on difficult specimens in our laboratory. We will also provide an update on tumor-specific markers as we review recent discoveries and discuss applications of different probe types as we present unique case studies. I'm going to start with reviewing the role that molecular cytogenetics plays in the identification of tumor-specific genetic events and some of the more recently identified genetic events that may provide diagnostic information. I will also discuss how you can best make use of the tissue resources available to provide the optimal specimen for the fish assay. Karyotypic changes were one of the earliest ways to differentiate and type solid tissue tumors. They often provide the starting point to guide the molecular studies to further define the underlying genetic mechanisms. The World Health Organization provides a classification series commonly used by pathologists on the histological and genetic typing of human tumors. This series of 7 books is more commonly referred to as the Blue Book series and provides a great reference for the genetics laboratory for understanding the molecular mechanisms underlying tumorigenesis of the different solid tissue tumors. In the previous edition of the WHO for classification of tumors of soft tissue and bone, less than a third of the tumor entries discussed recurrent cytogenetic and/or molecular abnormalities. However, 10 years later, when the fourth edition was released, we see several new additional neoplastic entities with an increase to over 45% with characterized cytogenetic and/or molecular abnormalities. Obviously, we have a lot more discoveries to make until the underlying genetic mechanisms for all tumors are known. New characteristic cytogenetic abnormalities with the corresponding molecular changes continue to be uncovered. You see here some of those more recently described since the latest edition of the WHO. Soft tissue angiofibroma is a relatively new histologic entity characterized as a benign fibrovascular soft tissue tumor of uncertain cellular origin. Jin et al. In 2012 reported that this tumor is characterized by an AHRR-NCOA2 fusion resulting from a translocation 58P15Q13. The evaluation of NCOA rearrangements using FISH could provide a practical method to confirm a diagnosis of soft tissue angiofibroma. In 2011, Trombetta et al. Described a translocation 719 Q22 Q13 as a sole abnormality in a case of pseudomyogenic hemangioendothelioma, and subsequently detected by FISH the presence of an unbalanced derivative 7, translocation 719, in an additional case. With the recent work by Walther et al. In 2014, it is now recognized that this translocation results in the fusion of the SERPIN1 and FOSB genes. Shimizono et al. Recently reported that NUP160 SLC43A3 fusion in angiosarcoma. Through a combination of FISH and RTP methodologies, they found 9 out of 24 angiosarcomas examined carried this fusion. Early studies suggested a more rapid progression of the tumor for patients harboring this fusion, with a significantly shorter duration between onset of symptoms and first hospital visit. There is frequent morphologic overlap between entities, and ancillary modalities are used in the vast majority of diagnoses. FISH is becoming increasingly important in the diagnosis of solid tissue tumors as an ancillary test. It is not uncommon for a pathologist to be presented with a case in which they do not have a clear idea as to the line of differentiation or normal cellular counterpart for a lesion. These cases certainly can present diagnostic dilemmas. In other instances, there can be significant overlap of the morphological features of some benign and malignant entities. There may also be considerable overlap in expression of IHC markers. For example, CD99, also known as MYC2, is commonly used as a marker of Ewing sarcoma. It may also be positive in lymphoma, synovial sarcoma, desmoplastic small round cell tumor, and rhabdomyosarcoma. While desmin, a common marker of muscle differentiation, is also seen in both rhabdomyosarcoma and desmoplastic small round cell tumor. It is also important to note that it is becoming more and more commonplace to make use of small biopsies for diagnostic purposes. Unfortunately, this often results in tissue not being available for conventional cytogenetic studies, so FISH may be the only opportunity to look at the genetic picture of a particular lesion. A very interesting study was presented by the group from the pathology department at the Brigham and Women's Hospital in Boston at the 2011 ASCO meeting. They performed a retrospective study to evaluate the rate of discordance between the histopathology diagnosis for challenging cases from referring institutions to their sarcoma center. They noted a discordance in the diagnosis in 24% of the 334 cases sent. 16% of the total cases resulted in a discordance that was deemed clinically impactful by either altering the diagnosis, the prognosis, or the treatment recommendations, including systemic therapy regimens, extensive surgery, or use of radiation. Some of the highest rates of discordance were seen with diagnosis in which FISH studies may provide diagnostic information, such as with the use of MDM2 or FUS and DDIT3 in various subtypes of liposarcoma, or FOXO1 or PAX3 or PAX7 in rhabdomyosarcoma. There are numerous options for establishing slides for FISH studies. You can perform FISH studies on slides from tissue cultures, on slides from cytological touch preparations from either fresh or frozen tissue, on unstained sections from formalin-fixed paraffin-embedded tissue, or even from tissue that has been embedded in OCT or optimal cutting temperature compound. This schematic hopefully illustrates the many options. We are fortunate in that we often receive fresh tissue for conventional cytogenetic studies. This allows us to have specimen available for FISH studies when the formalin-fixed embedded sections are not available due to the exhaustion of tissue with numerous IHC or other staining procedures, or in instances where the fixed tissue is not amenable to, or when the FISH tissue is not amenable to FISH studies, such as decalcified specimens. It has been our experience that the success of decalcified specimens for FISH is tissue-specific and may be only successful in about half the time. There will be times when it is most appropriate to FISH a destained metaphase preparation, such as when you have complex karyotypic findings with marker chromosomes or additional material of unknown origin. Interface FISH studies may yield variant signal patterns that lead to misinterpretation of the karyotypic abnormalities in these instances. Conversely, interface FISH studies may yield the appropriate abnormal results when the neoplastic cells are not proliferating in culture, or particularly when tissue is not submitted for conventional cytogenetics. For our laboratory, we have found that the FISH preparations generated from the single cell suspensions of enzymatically disaggregated tissue is optimal to traditional touch preparations generated from touching a solid tissue piece to a slide surface. FISH studies on unstained sections from formalin-fixed embedded tissue are particularly helpful in isolating neoplastic cells from normal contaminating stromal cells in heterogeneous samples. FISH on precipitating fixatives other than formalin, such as B5, have been successful, but may require an optimization of the pretreatment steps that varies from those that your laboratory uses for formalin-fixed tissue. We are fortunate that we have not had to work with many B5 samples in our laboratory, but if you find yourself in the situation where you are struggling with these specimens and are not finding success, you may want to review the manuscript by Schurter et al., where they describe the methods they used for various tissue types. For their protocol, they found the best results on the B5 fixed tissues when they increased their pepsin digestion time and reduced their proteinase K digestion time. G-banded slides that were analyzed for conventional cytogenetics may be destained and subsequent FISH studies performed. This is also useful when you want to FISH on the same metaphase that was karyotypically analyzed, specifically when you want to confirm any subtle or questionable abnormalities. We routinely use right stain for our G-banded preparations, which can be removed relatively easily with ethanol and 3:1 methanol to acetic acid washes. We follow this with a brief incubation in 2 times SSD and sequential dehydration steps with increasing concentrations of ethanol. Please note, though, that cultured cells may often not be the preferred specimen for FISH studies. Unless you have identified the presence of an abnormal clone, the culture cells may represent an overgrowth of normal fibroblasts and thus no longer contain neoplastic cells. For tissue culture purposes, solid tumors can be placed in 3 general categories. 1, those that grow attached to a substrate as a monolayer with cell-to-cell concentrations or connections, those that are anchorage independent and grow in suspension, and those that may have cell populations in both states. What is presented here is a partial list of those categories based on our experience with various tumor types and which cultures more often yield abnormal results. That is not to say that you will never capture neoplastic cells in the monolayer cultures of neuroblastomas or in the suspension cultures of undifferentiated pleomorphic sarcoma. This is just a list that is proven to be optimal for our laboratory. It is important to establish appropriate culture types dependent on the growth habits of the specific tumor. The category you choose for a specific tumor is not dependent upon the anatomical site, but the tumor type or diagnosis. There are numerous tumors that present diagnostic dilemmas, and you never know when one such case will arise. If you are receiving fresh tissue for conventional cytogenetics, it is important to be prepared for the possibility of subsequent FISH studies. You can do this by snap freezing a small portion of the tumor whenever it is logistically possible. This may mean that you do not set up as many cultures as you would traditionally if the tissue sample is small, but in many instances, having that tissue available for FISH studies is very impactful for patient care. If you do not feel you have adequate tissue to allow for a snap freeze, you can make touch preparations and store them at -20°C for later use. Following a fixation step with 3:1 methanol to acetic acid, they can be stored at -20 for several years and still be acceptable for FISH studies. If we are presented with a tissue that has numerous diagnoses in the differential, and particularly if there is an uncertainty that it represents a solid tissue tumor versus a lymphoid process, we will definitely set up both monolayer and suspension cultures. In our laboratory, we have made it a policy to collect the media from the first culture change on the in situ cultures and to harvest it as you would a traditional suspension culture. This becomes a very important culture in some instances, particularly when you are presented with those cases in which the tissue size is very limited, but the differential is extensive. So what I hope I showed you is that FISH studies can teach us a lot about the underlying genetic mechanisms of solid tissue tumors, and that there are many factors in the initial assessment of the tissue received that will influence the ability to obtain informative results. While FISH is a low-throughput cytogenetic test, its specificity, simplicity, and reliability continue to make it an integral component to a genetics laboratory, and it plays an increasingly important role in detecting tumor-specific biomarkers. Now it is my great pleasure to turn this discussion over to Pam Althoff.
Thank you, Mary, for that informative overview. In the remainder of the webinar, I would like to discuss the different types of probes utilized in FISH testing on solid tissue tumors, review some of the specific types of solid tissue tumors and their associated genetic abnormalities, and highlight some key scenarios and situations that should be considered when analyzing these cases. However, first I would like to talk briefly about probe selection and familiarity. When bringing new probes into the laboratory, it is important to consider the design of the probes available from different vendors to ensure that they properly cover all variants of the abnormality that you are intending to detect. If it is clear that a probe does not allow for the detection of all reported variants and there is not a more comprehensive alternative available, then a disclaimer should appear on the report. Also, familiarity with each probe's performance should be established during the validation process. Remember, not all probes are created equal, and each will have their own characteristics and nuances. While there are an ever-increasing number of commercial solid tissue tumor FISH probes available, it may be necessary to develop your own custom FISH probes in order to provide a more comprehensive solid tissue tumor test menu. The development of custom FISH probes is an entire webinar in itself, but here we highlight a few key questions that should be asked when producing custom FISH probes. The key to proper cone selection and probe design is a comprehensive understanding of the abnormality you are trying to detect and the regions of the genome involved. I will begin our discussion about specific probe types and their usage in the detection of abnormalities in solid tissue tumors by talking about FISH testing for copy number changes. Probes intended for detection of copy number changes are typically designed with a probe covering the gene or region of interest and a probe located elsewhere on the chromosome. Often, the gene or region-specific probe is labeled in the stronger fluorochrome, as that probe is typically smaller than the probe utilized as a control. As I am sure we are all familiar with, a normal signal pattern would consist of 2 red and 2 green signals that correspond with 2 copies of the region of interest and control probe respectively. These probes can be utilized to detect heterozygous deletion, homozygous deletion, or gain of the region of interest with or without concurrent abnormality of the control probe. Abnormality is typically determined by an abnormal cutoff established during the probe validation process. These probes can be utilized to detect gene amplification of the region of interest also. Although this abnormality may also be defined by an abnormal cutoff established during validation, often amplification is described as a ratio of number of signals for the region of interest to the number of signals for the control probe. The first disease I will discuss that is genetically characterized by copy number change is neuroblastoma. Neuroblastoma is the most common extracranial solid tumor of infancy. It is an embryonal malignancy of the sympathetic nervous system arising from neuroblasts. Neuroblastoma accounts for approximately 7.8% of all childhood cancers. It is the most common solid tumor in children under the age of 1 and the most common solid tumor arising outside of the brain in children of all ages. Age, stage, and biological features observed in tumor cells are important prognostic factors and are used for risk stratification and treatment assignment. Patient outcomes vary considerably from spontaneous regression in infants to disseminated chemo-resistant disease with a fatal outcome. Patients that fall into the low risk and intermediate risk categories tend to have an excellent prognosis and outcome. However, those with high-risk disease continue to have very poor outcomes despite intensive therapy. Unfortunately, approximately 70 to 80% of patients older than 18 months present with metastatic disease, and less than half of these patients are cured. Amplification of the MYC-N gene at 2p24 is observed in approximately 25% of patients and has been identified as an independent prognostic factor in patients with neuroblastoma. MYC-N amplification has been associated with rapid tumor progression and poor prognosis in children of all ages. On a conventional karyotype, MYC-N amplification is typically observed in the form of homogeneous staining regions or double minutes. FISH assessment is performed utilizing a ratio of MYC-N signals to chromosome 2 control signals. A specimen is considered abnormal when the ratio of MYC-N signals to chromosome 2 control signals is greater than or equal to 4. As you can see in the table, higher levels of amplification, specifically ratios greater than 10 and greater than 30, are listed separately, as studies have shown an inverse correlation between overall prognosis and increase in ratio. To note, cases that exhibit a ratio between 2 and 4 are interpreted as gain, not amplification of the MYC-N locus. Next, I would like to discuss the use of copy number probes for assessment of lipoma and atypical lipomatous tumor and well-differentiated liposarcoma. For simplicity, I will exclusively use well-differentiated liposarcoma for the remainder of this section. Lipoma is a benign mesenchymal tumor that is composed of mature adipocytes that may closely resemble normal fat. Lipomas are the most common mesenchymal neoplasm in adults, and they rarely reoccur and have no risk of progression. In some cases, well-differentiated liposarcomas may resemble lipomas. However, they tend to be larger, have adipocytes that show greater variation in size than an ordinary lipoma, and the presence of lipoblasts may be observed. Well-differentiated liposarcomas located in surgically amenable locations do not tend to reoccur after complete resection. However, local recurrence is not uncommon. Deep anatomic sites, like retroperitoneal lesions, tend to recur. These lesions may cause death from uncontrolled local effects, and they may dedifferentiate and metastasize. Distant metastases are observed in 15 to 20% of cases where dedifferentiation occurs, with an overall mortality of 28 to 30% at 5 years. As there are histologic similarities between lipoma and well-differentiated liposarcoma, the distinction between lipoma and well-differentiated liposarcoma may pose a diagnostic dilemma. Cytogenetically, well-differentiated liposarcomas are characterized by the presence of supernumerary ring chromosomes composed of amplification of 12q13 to 12q15. By FISH, this abnormality is often identified by assessing for amplification of the MDM2 gene locus. As with MYC-N amplification in neuroblastoma, MDM2 is often assessed as a ratio of MDM2 signals to chromosome 12 control signals. However, MDM2 is considered amplified in cases with a ratio greater than or equal to 2. Given the overlap in adipocytic lesions, laboratories may be running FISH probes for abnormalities observed in lipoma or myxoid liposarcoma, such as rearrangement of the HMGA2 or DDIT3 genes, respectively. It is important to be cognizant that these loci are often present in the supernumerary ring chromosomes observed in well-differentiated liposarcoma cases. So, although you may not have been initially assessing for well-differentiated liposarcoma, be aware of possible amplification patterns, as shown in these two examples on the right, when utilizing these probes covering genes like HMGA2 or DDIT3. These findings are more suggestive of well-differentiated liposarcoma, and therefore they should be reported as such. Rhabdoid tumors are highly malignant tumors, mainly affecting infants and children. This group of morphologically and genetically identical tumors include atypical teratoid rhabdoid tumors, which present in the brain and central nervous system, malignant rhabdoid tumor of the kidney, and extrarenal rhabdoid tumors. The overall prognosis for these patients is extremely poor, regardless of location. Although patients older than 3 years of age have a slightly longer survival, most likely due to the ability to use more aggressive treatments, the overall survival, 5-year survival, is less than 15%. Approximately 98% of rhabdoid tumors exhibit homozygous inactivation of a classic tumor suppressor gene, SMARCB1, located at 22q11.2. Sequencing mutations, partial and whole chromosome deletions, and copy-neutral loss of heterozygosity of SMARCB1 have been described. Approximately 35% of patients have germline mutations of SMARCB1. In cases with one mutated allele, the second allele is often lost by deletion or mitotic recombination. An example of this is seen in the image on the top left. In addition, homozygous deletions are frequently observed in rhabdoid tumors. When assessing for homozygous deletion, be sure that proper controls are in place to ensure that this finding is the result of tumor pathogenesis and not a poor quality hybridization. This can be accomplished internally by ensuring appropriate hybridization quality on accompanying normal cells, or externally with a known normal control that is run concurrently. The image on the bottom left shows an area with homozygous deletion and an area of normal tissue where the red SMARCB1 probe is present. As we are discussing the assessment of dilution, I would like to discuss a scenario that highlights the importance of correlating fish findings with adjunct studies such as conventional cytogenetics, as well as the importance of utilizing control probes in situations where whole chromosomal loss is being assessed. For example, a specimen is received into the laboratory with a clinical indication such as meningioma, where the characteristic genetic finding is complete loss of chromosome 22. Interphase studies on cytologic touch preparations from the tissue received for conventional cytogenetic studies reveals 2 copies of 22q. However, cytogenetic analysis revealed an abnormal hypertetraploid clone characterized by loss of 22. Without the concurrent conventional cytogenetic analysis, this case would have been improperly called negative, as the fish signal pattern indicated this to be the case. There are other measures that can be employed to assist in scenarios like these. Specifically, the addition of a separate control probe, such as the chromosome 4 centromere in this instance, may suggest the presence of an abnormal polyploid population exhibiting loss of the region of interest. However, this alone may not be a definitive assessment. If appropriate material remains, running additional centromere probes may allow for additional discernment about the ploidy status of this particular specimen. Although gain of multiple centromere probes may not be a conclusive confirmation of ploidy status of the sample, it might prompt a more conservative interpretation in light of the suspected diagnosis. We will now shift our discussion to the use of dual color break-apart probes in the assessment of abnormalities in solid tissue tumors. Dual color break-apart probes are designed to detect rearrangements of a particular gene or region of interest. These probes are designed with a probe labeled in green that maps to one side of the region of interest and a probe labeled in red that maps to the opposite side of the region of interest. In normal cases, when the gene is intact, the probes will be juxtaposed, resulting visually in a yellowish signal. When the gene is disrupted due to rearrangement of the region, the red and green signals on the affected homolog will separate. Although dual-color break-apart probes are useful diagnostic tools, it is important to remember that you cannot identify the gene partner involved in the rearrangement. Additional probes will be necessary to identify rearrangement partners in cases when that information is of clinical relevance. Synovial sarcoma is a mesenchymal tumor with varying degrees of epithelial differentiation that accounts for 5 to 10% of all sarcomas. While synovial sarcoma may occur at any age, it is most commonly observed in adolescents and young adults with a median age of 30. Majority of tumors arise in the deep soft tissue of the upper and lower extremities, followed by the trunk and then head and neck regions. Prognosis is based on tumor stage at presentation, tumor size, grade, and the initial care received. Patients with small tumors that can be completely resected have a very good prognosis. In addition, children have a better prognosis than adults. Genetically, synovial sarcoma is characterized by a translocation between the long arm of chromosome 18 and the short arm of chromosome X, which is observed in greater than 95% of synovial sarcoma cases. This abnormality results in disruption of the SS18 gene at 18q11.2 and subsequent fusion of SS18 to one of the SSX genes. SSX1 fusions occur in 2/3 of cases, SSX2 fusions occur in 1/3 of cases, and SSX4 fusions have been observed in a few rare cases. At this time, there is inconsistent evidence about the clinical value of the different reported transcripts. Ewing sarcoma is a relatively rare primary bone tumor, accounting for only 6 to 8% of primary malignant bone tumors. However, it is the second most common bone and soft tissue tumor in children and adolescents, with approximately 80% of cases occurring before the age of 20. Advances in treatments have improved considerably, with a 5-year survival rate of 54 to 74% in cases that present with localized disease. Unfortunately, patients that present with metastatic disease still have a very poor prognosis, with a 5-year survival rate of only 10 to 35%. Ewing sarcoma is genetically characterized by recurrent balanced translocations involving the EWSR1 gene and one of several members of the ETS family of transcription factors. This finding is observed in approximately 90% of patients with a clinical diagnosis of Ewing sarcoma. It is important to note that rearrangement of the EWSR1 gene has been described in a wide variety of tumor types, as is illustrated in the table here. In addition, some of these different entities may fall into the differential diagnosis with Ewing sarcoma. Therefore, if multiple tumors with known rearrangements of EWSR1 are being considered in the differential diagnosis, additional FISH testing with probes that assess for some of these common disease-specific gene partners may be warranted. When utilizing dual color break-apart probes, it is not uncommon to encounter variant signal patterns that may require additional testing and consideration. One common occurrence may be loss or gain of one portion of the probe, as is illustrated in the images here. The image on the top right exhibits what appears to be loss of the 3' centromeric ETV6 region probe, while the image on the bottom right represents what appears to be gain of the 5' telomeric ETV6 region probe. However, due to the design and limitations of the probe utilized, it cannot be determined whether these findings represent rearrangement of the ETV6 gene region or loss or gain of chromosome 12 material. If common gene partners are typically associated with the entities that fall into the differential diagnosis, follow-up with available dual fusion probes may allow for more complete characterization of these signal patterns. Another option, if available, would be to perform sequential FISH studies on abnormal metaphases from the conventional cytogenetic studies. This may be of particular use in cases where the aberrant signal pattern is of clinical significance. Let's look at the following example. A translocation between the short arm of chromosome 12 and the long arm of chromosome 15 has been observed in patients with a clinical diagnosis of infantile fibrosarcoma, as well as in patients with mammary analog secretory carcinoma and secretory carcinoma of the breast. This translocation results in fusion of the protein dimerization domain from the 5' region of the ETV6 gene with the protein tyrosine kinase domain from the 3' region of the NTRK3 gene. This fusion leads to constitutive tyrosine kinase activity. When utilizing an ETV6 dual color break-apart probe to assess patients for this abnormality, signal patterns exhibiting loss of the green 3' probe resulting in single red signals should grab your attention. This signal pattern may represent an activating deletion, as the dimerization domain of ETV6 is present with the remaining 5' portion. Performing studies on de-stained metaphase cells can assist in confirming this finding, as is the case in the example here, where the remaining 5' ETV6 signals are present on the derivative chromosome 15s identified in this patient's concurrent G-band study. The final type of probe we will discuss today are dual fusion translocation probes. These probes are intended to detect translocations involving 2 genes or regions of interest. Dual fusion probes are designed with a probe labeled in green that spans the first gene of interest and a probe labeled in red that spans the second gene of interest. As with copy number probes, a normal signal pattern is the presence of 2 red probe signals and 2 green probe signals that correspond with 2 intact copies of each loci. When a translocation occurs, disruption and subsequent translocation of each loci results in the presence of 1 green signal representing an intact copy of gene A red signal representing an intact copy of gene B, and 2 yellow fusion signals that presumably represent 1/2 of the translocation. Dermatofibrosarcoma protuberans, or DFSP, is a rare soft tissue tumor accounting for approximately 1% of all soft tissue tumors. DFSP is a superficial, low-grade dermal tumor that occurs most frequently in the trunk, proximal extremities, and head and neck regions, and it affects young to middle-aged adults. Local recurrence occurs in 20 to 50% of patients, often due to a lack of wide margins. Approximately 10 to 15% of patients will experience high-grade fibrosarcomatous progression. Although metastasis is usually rare, distant metastases are observed in approximately 13% of patients with high-grade fibrosarcomatous progression. DFSP cases are cytogenetically characterized by a translocation, often in the unbalanced state, between the long arms of chromosome 17 and 22, which results in fusion of the COL1A1 gene at 17q21 with the PDGF beta gene at 22q13. This fusion ultimately leads to the activation of the PDGF receptor tyrosine kinase, triggering the proliferation of DFSP tumor cells. Patients with this abnormality may be eligible for targeted therapy with a tyrosine kinase inhibitor. Given the likelihood that this translocation will occur in an unbalanced state, variant and often single fusion signal patterns are common, as is illustrated by the destained metaphase image shown here. NUT midline carcinoma is an extremely rare, highly aggressive tumor that arises in the midline of the body. NUT midline carcinoma patients often present with locally invasive, widespread metastatic disease, and the outcome is dismal with a median survival of 6 months. NUT midline carcinomas arise from various anatomical sites and therefore cannot be characterized by the same system as most other carcinomas that are defined by the tissue of origin. Instead, NUT midline carcinoma is defined genetically by the presence of chromosomal rearrangements involving the NUTM1 gene at 15q14. 2/3 of patients with NUT midline carcinoma are characterized by fusions of the NUTM1 gene with the BRD4 gene on 19p13.1, which results in a recurrent gene fusion resulting in cellular epigenetic deregulation. The remaining 1/3 of patients are characterized by NUTM1 fusions with a gene other than BRD4. When utilizing a dual fusion translocation probe, these cases would present with 3 signals for the NUTM1 probe, represented as 3 green signals in the image on the bottom left. With this assay, you would be unable to determine whether this finding represents rearrangement of the NUTM1 gene or multiple copies of 15q14. Subsequent studies utilizing a NUTM1 break-apart probe would be essential in ascertaining this information. However, if a break-apart probe was not available for this region, a comment or disclaimer about the inability to further characterize this finding is recommended. Today, we provided an overview of the approaches that are commonly utilized in the cytogenetic laboratory in the assessment of FISH abnormalities in solid tissue tumors, including how FISH plays a role in the identification of tumor-specific genetic events, and some of the more recently described genetic abnormalities that may play a role in the diagnostic arena. Also, we aim to provide some helpful tips to best use the tissue resources that are available for testing. In addition, we hope that you are more familiar with the different probe types and applications of these probes in solid tissue tumor genetics. Finally, we introduced some common situations and scenarios that are encountered when performing FISH analysis on solid tissue tumors. Mary and I have the good fortune to work with a wonderful and skilled group of staff and technologists here at the Human Genetics Lab at UNMC. We would like to acknowledge the FISH and tumor teams for their continued dedication to the assessment of these challenging cases. We would also like to take a moment to thank our laboratory directors for their mentorship and support. Finally, a special thank you to Nicole Hackendahl for her valuable assistance in making this presentation possible.
Thank you, Pam and Mary, for a tremendously informative presentation. We appreciate you both sharing your expertise with us today. I will just reiterate that in the lower portion of that panel that you see on the right-hand side of your screen, you can type in a question or comment and submit it to the webinar organizers at any time. So please, if any questions have come to mind over the course of the presentation, please feel free to submit those questions and we will try to get them answered over the next 10 minutes or so. And I think with that, I'll start with the first question, which is that, I think some laboratories struggle with the fresh tissue versus paraffin, what tissue to be fishing, and how to really make that decision. Can you guys speak a little bit to your experience with the fish on fresh tissue versus on paraffin-embedded tissue?
Well, Jennifer, it has been our experience here at the laboratory that when there is paraffin tissue submitted by the pathologist, we often refer to that first. Hopefully they have marked on the H&E and on the unstained section exactly where they have localized the tumor cells. So that allows us really to focus our studies there. Like I said, sometimes with the cultured cells, you do unfortunately come across those instances where the normal fibroblasts, the stromal cells that are contaminating the tissue have overtaken the culture, and that's really basically all you're left with. So you might be just fishing just normal cells. However, you know, there are certainly times, like I said, when paraffin was unable to be submitted for various reasons, or if the paraffin doesn't really make for a good fish study. Like I said, decalcified studies, decalcified tissues can sometimes be problematic. Also, sometimes, you know, we have a great success rate with our unstained sections. Pam's group has really done a tremendous job, but there are those occasional specimens that just don't allow for a successful fish assay. So to be able to go back to maybe some of that snap frozen tissue or touch preparations that have been made previously and really represent the original tumor are a great resource. But there still are those instances where you have those really questionable cytogenic abnormalities and you suspect something that's actually outside of what's in their initial differential. You're just really curious, does that, what appears to be a translocation between 12-15, does that really involve ETV6, and that's outside of their differential? So you may want to go to those specific abnormal cells to answer those type of questions. So you can really see that pattern on those abnormal karyotypes instead of being misled with any variant patterns that are in your interface study.
Excellent, thank you. One of the questions that came in from an attendee is the question, does your lab make their own probes or use vendors for custom probes of targets that are less readily available in the catalogs?
Yes, majority of the probes we're currently utilizing as part of our clinical testing menu are commercially purchased probes from one of the different various vendors. However, when probes are not available through commercial vendors, or the probes that are available don't meet the standards that we would like to utilize for our clinical testing menu, we do have a department here in-house where we make our own custom fish probes, particularly for those diseases or disorders that are extremely rare, and commercial vendors don't find it advantageous to produce probes that cover those types of abnormalities.
Excellent. Another question, how do you deal with the expiration of probes that might be used very rarely on these more rare tumor types?
Well, honestly, in our ability to stay compliant with the CAP regulations, once fish probes have reached their expiration date, they are cycled out of the usage for clinical testing purposes. We do maintain these probes within our lab for research projects and research-driven testing, but from a billable clinical testing standpoint, we remove them from our clinical testing menu.
Great, thank you. There's also a request for some more tips on how to improve fish after G-banding. The attendee is commenting that they see very mushy chromosomes and kind of diffuse fish signals on those destained metaphase spreads. So any advice that you might be able to offer on how to kind of crisp up those signals?
Absolutely. I do think, and Mary touched on this a bit, the type of stain used in your G-banding procedure may impact the ability to get that stain, remove that stain from the preps and be able to have nice, clean, crisp hives. If you don't get all that stain out, you end up with this, those of you that have done this horrible green wash over the slide that makes it impossible to do a really thorough, appropriate assessment. We use rights here in the lab, and so we've had good luck with being able to remove that from the cells. We have a protocol where we do destain, take the oil off with the hemo-D and then destain with ethanol and methanol acetic acid fixed steps, followed by 2-XSSC pre-treatment and a warm bath. And then we are able to go ahead and use our standard denaturation and hybridization protocols that we would typically utilize for fresh dropped samples with a great deal of success.
Great, thank you. Another question, just about the laboratory in general that you come from. Is it exclusively a cancer lab, or do you do prenatal and congenital testing as well? So I think if either of you could speak a little bit to the scope of the laboratory that you come from, that would be great for our listeners.
Well, I could speak to the makeup of fish specifically. We do both, assess both cancer and constitutional abnormalities in fish. Our caseload typically is approximately 80% of hematology, oncology, or solid tumor testing, with the remaining 20% approximately falling into the prenatal and postnatal arena. The lab as a whole is a full-service cytogenetics laboratory offering tests, both conventional, we offer conventional cytogenetics, fluorescence in situ hybridization, as well as microarray and next-generation sequencing, all that cover both neoplastic and constitutional abnormalities. And also we are doing testing within the hereditary cancer environment as well.
Excellent, thank you. Does your laboratory use an off-the-shelf tissue pretreatment kit or something that you've homebrewed?
We are currently doing pretreatment utilizing the VP2000 processor, and we are utilizing the reagents that can be purchased through Abbott Molecular at this time.
Do you have any problems with bacterial contamination of your right stain?
No, we've been really fortunate. We really have not encountered that at all. Actually, years ago when we were using the Geam sustain, that tended to be a little bit more problematic because we were using various buffers to kind of make that staining process maybe just a little bit more gentle. But for our right stain, and honestly, we actually probably go through it fairly quickly, that it being contaminated with bacteria has not been an issue for us.
Knock on wood, right? Excellent, thank you. Another question about fishing the fresh tissue. Do you prefer touch preps or a direct harvest for fish?
I do believe that for us, we have the best success with making that kind of touch preparation sample from our single cell suspensions after we have enzymatically disaggregated our tissue. It's really been quite useful in the fact that we really waste no tissue at all, because we're not having to make those fresh cuts on that original sample and kind of making those gouache preparations as you would as you were applying that, pressing that tissue piece on your slide. But it allows us to, after we've done our enzymatic disaggregation, you're pulling that sample up into your pipette, and invariably, you get a little bit of cells stuck in that very bottom of your pipette tip. And so by just touching that to your slide surface, that allows for that to be pulled out of that pipette, and it makes a very nice touch preparation. And if you're doing a direct harvest, you may find that you don't have the number of cells that you would really like to capture there. Sometimes, like I said, we're trying to set up both in situ and suspension cultures, so sometimes we're having to really triage very small samples, and those suspension cultures can become quite small. Sometimes we'll even do half cultures. But as you're spinning that out and doing your harvest, you do have the potential of reducing the number of cells you have.
And kind of to comment a little bit too on the use of the disaggregated cells versus the traditional tissue touch preps. The fish signals are remarkably better on those disaggregated cells. They're easier to interpret, they're not as diffuse, and they're not as strung out as they are on those traditional touch slides. And we haven't seen a difference in the ability to detect the abnormalities we're trying to find. So it's been a really useful tool in our lab to circumvent some of the problems that you run into with low tissue volumes and kind of the sketchy signals that you might get on a traditional touch prep.
Great. I think The one last question before we wrap up today would just be, do you have any kind of tips on how to improve your hybridization quality on paraffins? What you guys typically do do you do any adjustments to your protocols for special tissue types?
We do run a slightly weaker pretreatment on lung samples that we are processing. We've also found, so we do have a couple of different pretreatment protocols that vary the time spent in the protease buffer, and we will make some modifications to the denaturation time. But mostly we've been fairly fortunate to put together a streamlined protocol that allows us to pre-treat all of our samples on one program and hybridize all of our samples on one program. One trick we have done is we are currently still utilizing, we're utilizing thermobrites for our denaturation and the beginning part of our hybridization process, but we've subsequently started moving our slides into a humidified incubator after we've reached our hybridization temperature, and we've found that that has been extremely helpful in reducing the need for rehybridization, and it's just made for a more efficient hybridization and higher quality signal.
Great, thank you both so much. And thank you all for participating in this webinar. Shortly, webinar participants will receive an email from AGT that includes a link to an evaluation. Please take a few minutes to access it and provide your feedback on today's session. The webinar today was recorded and will be posted on the AGT website in the near future. We hope that you will join us for upcoming webinars over the next few months. We will send out a notification to members once registration is available for our next webinar. Also, we hope to see you all at AGT's annual meeting in Orange County, California, which is scheduled for June 23rd through the 25th in 2016. The meeting organizers have been working diligently to put together a balanced program of both cytogenetic and molecular genetic topics. Registration will be open in early 2016, and we hope to see you there. Again, thank you.